Hello everyone, welcome to today's session on endoplasmic reticulum. The endoplasmic reticulum or ER is a large membrane bound structure inside the cell. It is directly connected to the nuclear envelope and spreads throughout the cytoplasm. The ER plays a key roles in protein synthesis, lipid metabolism, calcium storage and membrane production. These functions make it essential for the cell's growth, communication, and survival. The endoplasmic reticulum is made up of a network of tubules and flattened sheets called cisternae. It exists in two main forms. The rough endoplasmic reticulum is covered with ribosomes, which help make proteins. The smooth endoplasmic reticulum has no ribosomes and is important for lipid metabolism, detoxification of harmful substances, and calcium storage. Together, these forms help the cell perform many vital tasks. The endoplasmic reticulum has three key structural parts. The tubules are narrow and dynamic, meaning they change shape and spread throughout the cytoplasm. The seeds or flattened cisternae are often found near the nucleus and help with protein synthesis. The junctions are three-way intersections that keep the ER network stable and connected. The endoplasmic reticulum acts as a cell's protein factory and quality control center. The rough endoplasmic reticulum or rough ER makes proteins that are destined for the secretory pathway, meaning these proteins will either leave the cell or go to the specific locations inside it. So, as these proteins are made, they are folded into their proper shapes with the help of a special helper proteins called chaperones, such as calnexin and calreticulin. If any protein is folded incorrectly or damaged, the ER identifies it and removes it through a system called ER associated degradation or ERAD. The faulty protein is retrotranslocated back to the cytosol, tagged with ubiquitin, and sent to the proteasome, the cell's recycling center. The smooth endoplasmic reticulum or smooth ER is mainly responsible for making lipids and sterols, which are important for building cell membranes and other functions. These lipids are then transported to different parts of the cell, either through small transport vesicles or directly at membrane contact sites, or called MCS. The ER also serves as a major storage site for calcium ions. Let's walk through one of the most elegant cellular communication system, calcium signaling via the ER. It all kicks off with a signal like a hormone or neurotransmitter binding to a receptor which produces IP3, a small molecular messenger. IP3 travels to the endoplasmic reticulum and binds to the IP3 receptor on its membrane like a switch. When this opens, calcium channels allowing calcium ions to flood into the cytoplasm. This surge of calcium sparks various cellular actions from muscle contraction to enzyme secretion. But calcium cannot linger freely for long. The ER needs to refill its store. Here, the sensor proteins STIM1 detects the drop in ER calcium. It quickly moves to the ER membrane areas close to the plasma membrane and activates ORAE1, a channel on the plasma membrane. This allows extracellular calcium to pour into the cell, recharging internal stores. Finally, sarcopoms step in and using ATP to transport calcium back into the ear, resetting the system for next signal. The endoplasmic reticulum has special regions called ER exit sites or ERES. These are areas without ribosomes where properly folded proteins leave the ER. The proteins are exit in small transport carrier known as copy 2 coated vesicles which taken them to the next stage of their journey, usually to the Golgi apparatus. The endoplasmic reticulum is highly dynamic, meaning it is constantly moving and changing shape inside the cell. This movement happens in three ways. First, motor proteins like kinesin 1, and dynein, which help pull and push the ear along the microtubules. Second, ear extends by attaching to the tips of the growing microtubules through tip attachment complexes or TSAs. Third is it can hitch like by linking to moving organelles such as endosomes and mitochondria and travel with them. So this constant motion helps the ear stay connected and functional. The endoplasmic reticulum forms special links with other organelles called membrane contact sites, MCs, I have told earlier. So these are close connections where the membrane come near each other, about 5 to 30 nanometer apart, but they do not fuse. Okay, so these contact sites help the ear transfer lipid to organelles like mitochondria, the Golgi, and the plasma membrane. They also play a key role in calcium signaling and in the splitting or merging of organelles during fission and fusion events. Let's explore how different organelles work together at membrane contact sites or MCS to keep the cell running smoothly. Mitochondria handle the exchange of lipids and calcium ions, which is essential for energy production and cell signaling. MCS are held together by tethering proteins like VAPB, PTPIP51, or protein tyrosine phosphatase interacting protein 51, and mitofusin 2, which bridge the gap between the two organelles without fusing their membranes. These sites serve as critical hubs for the communication. One major function is lipid transfer, helping maintain membrane composition on both sides. Another vital role is signaling, where the calcium releases from the ear through the IP3 receptors is handed off to the mitochondria via GRP75 or glucose regulated protein 75, which is also called mortalin, and VDAC1 channels like voltage dependent anion channel 1. Importantly, these contact points also act as the initiation site for mitochondrial fission. Here, the protein DRP1 is recruited and facilitates division of the mitochondria, which is essential for maintaining mitochondrial health, distribution, and function. In short, these contact sites coordinate metabolism, energy production, and organelle remodeling, a perfect example of inter-organelle teamwork in the cell. Key proteins like VAPs or VAMP associated proteins, MFN2 or mitofusin 2, and PDZ-D8 or PDZ domain containing protein 8 help tether mitochondria and facilitate this transfer. The plasma membrane is involved in store operated calcium entry or SOCE and lipid transfer. Proteins such as STIM1 or stomatal interaction molecule 1, ORAE1 or calcium release activated calcium channels protein 1, or E sites or extended synaptotagmins, which manage calcium sensing and lipid movement to maintain the cellular health. Endosomes support their maturation and move cholesterol between membranes, relying on RAB7 or RAS related protein RAB7. VAP proteins or VAMP associated proteins and ORP1L or oxyterol binding protein related protein 1L to regulate these tasks. Finally, lipid droplets grow and form through the action of sapin and RAB18 or RAS related protein RAB18, which help manage fat storage and energy balance. The endoplasmic reticulum or ear is vital for many cellular functions. But when its shape or dynamics go wrong, it can lead to serious diseases. For example, neurodegenerative disorders like amyotrophic lateral sclerosis or ALS and hereditary spastic paraplegia or HSP are linked to ER dysfunction. In these conditions, the ER fails to maintain its proper structure, which disrupts nerve cell health. Metabolic diseases such as diabetes, which are also connected to the ER problems, especially when the ER cannot properly handle protein folding or lipid metabolism. Some pathogens, including bacteria like Legionella or the viruses like Zika, hijack the ER to support their replication, further damaging the cell. Central to many of these diseases is ER stress, when the ER is overwhelmed by misfolded proteins. So, this activates a protective mechanism called unfolded protein response or UPR. While UPR tries to restore normal function, prolonged ER stress and UPR activation can contribute directly to disease progression also. The endoplasmic reticulum or ER is a highly versatile organelle, essential for cell survival, specialization. Its unique shape and constant movement are closely connected to how well it performs its function. Through membrane contact sites and dynamic remodeling, the ear interacts seamlessly with other organelles, integrating itself into the cell's entire network. So, this intricate coordination is what enables the cell to function efficiently and respond to changing conditions. So, if you enjoyed this session and found it informative, please don't forget to like this video, share it with your friends and subscribe to the channel so you can stay updated on the future content. Thanks for watching.